Welcome back to the Classics of Immunology Journal Club. I want to just remind you, if you really like this journal club and you like the video, please like it on the YouTube website and click on the little bell to get notified when I put a next one on. And also scroll down and go to my website, kendallasmith.com. There's all kinds of things there, including and importantly, all of the classic papers in immunology are on that website. So you don't even have to go to your library. You got them in the Kendall A. Smith Library. So today we are going to review two papers from 1972 that really became the classic papers of what was called lymphocyte-activating factor, or LAF, which became interleukin-1. Took a while, about a decade later. But this is the beginnings of the mitogenic factors found to be released by macrophages. So the first paper is entitled The Potentiation of T Lymphocyte Response to Mitogen. And this is number one in this series, The Responding Cell. And the authors of this are Yigal Gary, Richard Gershon, and Byron Waxman. Now, Yigal Gary was an Israeli scientist who was on leave of absence from Hebrew University, and he had found his way to Yale, where he hooked up with Waxman's group. Waxman was the professor, and Dick Gershon was a junior faculty person at that time. And Waxman was famous because of his father won the Nobel Prize, Selman Waxman, for discovering streptomycin, which was the first antibiotic that was effective against tuberculosis. In the introduction of this paper, the authors cite several papers that macrophages are necessary for the antibody response to sheep red blood cell. And they referenced Don Mosher's paper that we reviewed, and Unanue's paper, and Hoffman and Dutton's paper. And then they go on and when they're doing Hoffman and Dutton's paper, they talk about the fact that there are some inklings that people have found soluble factors that the macrophages might make that would potentiate the response of mice in antigen-specific responses and mixed lymphocyte cultures, and also to make antibodies to red blood cells. But this paper is about the so-called non-specific mitogens, and you may recall that we started the cellular immunology era reviewing Peter Knowles' paper, who discovered accidentally that phytohemagglutinin was a mitogen, and then subsequently, once T cells were discovered, only in 1969-70, it was then pinned down that PHA for human and mouse lymphocytes only stimulated T cells. The other thing that they reference that's really, I think, very important and interesting is the fact that macrophages were thought to play a role in the action of immunological adjuvants. Now, you may remember that we sort of started way back in the early days of in the late days of the 19th century with William Coley, who was giving bacterial extracts, dead bacteria, to cancer patients to try to boost up their reaction, their hopefully anti-tumor reaction. And so his experiments were really the first descriptions of what became to be known as immunological adjuvants. And of course, it then was carried on in the 1940s with the discovery of the fact that TB organisms would potentiate antibody or immune responses, particularly cell-mediated immune responses, and so forth. So the question was, how does that work? What's the mechanism of action of adjuvants? This paper is one of the first to be able to try to dissect that question. They state in the introduction that this paper shows that macrophages release mitogenic substances, and that those activities work on T cells, and they also potentiate the response to nonspecific mitogens like PHA. And furthermore, and lastly, that their production of these mitogenic factors that they say are coming from macrophages, and that's in the second paper, is markedly potentiated by macrophage stimulants, in particular, lipopolysaccharide, bacterial endotoxin. And so all of these things are very, very important for what came before and also what came subsequently in the whole dissection of the cellular immunological approach to the immune response. So in the materials and methods of this paper, they use mice to get thymocytes and spleen cells and bone marrow cells to study. And they also drew blood from their lab mates to get human peripheral blood mononuclear cells to study. They separated particularly the thymocytes on discontinuous bovine serum albumin gradients. And so they would have different concentrations of bovine serum that they would layer into a centrifuge tube, and then they would put on top the cells, and then they would spin those cells, and they would layer out depending on their density. So they had they were able to get 4 different bands there, and which they just labeled A, B, C, and D, and then P, which was the precipitate in the bottom of it. In order to get so-called macrophages or adherent cells to study, they used the trick that we've talked about before of simply putting the cells down on culturing them in plastic flasks or glass flasks, and the adherent cells would adhere. And then you could wash off the non-adherent cells, mostly lymphocytes, and then you'd have a pretty pure culture of adherent cells. They also used nylon wool columns to separate, particularly when they were dealing with human lymphocytes. You could put nylon wool into a syringe. We used to use a plastic syringe, 10 cc syringe, about that big. You can also use a Pasteur pipette. You can put that in the autoclave and sterilize it, and then let it cool down. And then you can put the cells on top and just let them, by gravity, come down through there, and then wash them down with medium. And the cells that make it through and don't stick in the nylon wool are basically, they're all lymphocytes. The adherent cells stay into the nylon column. So that when they cultured their cells, and most of this is with mouse cells, they cultured them at a pretty high density, 5 million cells per ml. For the thymocytes, and for the splenocytes, it was 2 million cells, 2 times 10 to the 5th cells per ml, 200,000. And they cultured these cultures for 3 days, and then they would then pulse them with radiolabeled thymidine, tritiated thymidine, for the last 24 hours, and then they could put the resultant gamish into the radioactive counter and get counts per minute, and that was their readout. Now, in order to stimulate the cells to incorporate thymidine, they used PHA at a suboptimal concentration. And that was very important because if they used an optimal concentration, their supernatants wouldn't potentiate their optimal response. So they used PHA, then they used also for the mouse, particularly they found that concanavalin A, which is another T cell specific mitogen that had been just recently shown to be a T cell mitogen. And they also used LPS at 100 micrograms per ml, and that's a really high concentration of LPS. In order to make their supernatants, they would culture the cells overnight, 24 hours, with either LPS or PHA, and then they would harvest the supernatant, spin out the cells, and then they would test them. So, and the results in this paper, I forgot to say that this paper was published in the Journal of Experimental Medicine, and the first and the second were back to back, and they became the classic papers describing what we subsequently called LAF. So first they tested the effects of human supernatants on mouse lymphocytes, both thymocytes as well as spleen cells. And they found that the supernatants induced by LPS onto the adherent cells markedly increased the thymocyte tritiated thymidine incorporation. They found that for the lymph node cells and the splenocytes, it didn't work quite as well. Now, the reason that thymocytes were important for them was because thymocytes are T cells, are T-derived, thymus-derived lymphocytes. And by this time, after Max Cooper and so forth in the 60s, this was now pretty much well accepted. And then with the recent experiments by Martin Raff of the theta antigen and the antibodies to theta showing that they could mark those and so forth, the thymocytes were pretty much accepted as pure T cells. You didn't add, that's number one. Number two, they didn't have very many macrophages. They had some, but not very many. And they didn't really basically have any B cells. And so this was a sort of a real simple way to get T cells. You just take the thymus rather than fiddle around with a spleen that's got a lot of macrophages and has a lot of B cells. God knows what else is in there, So they showed in their paper and in one of their figures that if they put the cells into culture and pulse them with thymidine after 1 day, 2 days, and 3 days, that there was spontaneous proliferation going on at 1 day, even without any PHA or any other stimulant or no supernatants and whatever else. And then that fell off over the next couple of 3 days. And so then if you added like PHA at time 0, it would come back up again. And so the counts per minute that they were then looking at were these sort of resurrected counts per minute in this system. They looked at a whole bunch of different mouse strains, about 4, 5, 6 of them, and they all worked fine. They all worked the same way. So there wasn't a problem. When they tried to fractionate the thymocytes, and this becomes interesting, over their bovine serum albumin discontinuous density gradients, they found that most of the very dense cells, which are the ones at the bottom 2 layers, and then also in the pellet, that accounted for 80% of the lymphocytes in the thymus. And those are the cells that really didn't respond well to PHA. And so it was the less dense cells that were in the upper layers that were really, and less than 20% of the total thymocytes where all the thymidine incorporation was happening. Now, nowadays we know that once the so-called T cell markers of CD4 and CD8 were discovered, we know that the less dense cells are the only ones that are immunologically mature. The very small dense ones, greater than 80% of them, are not mature yet, and they can't proliferate into mitogens and so forth the way the mature ones do. So that subsequently happened to be proven later on, but these guys found it early in this particular paper. When they took splenocytes and they purified out the macrophages, the adherent cells, and then added back the supernatants and so forth, then they could get a response. And so the purified T cells from the periphery, as well as the purified T cells from what they call central T cells from the thymus, worked. So those were the data that they reported in this paper. And in the very first paragraph of the discussion, they state that these data establish that the proliferative response of T cells to mitogens is increased by soluble factors. So that's a pretty solid statement. Whether they could really say that at that particular point in time or not, without really knowing what the factor was, is another question, but that's what they say. And they proposed the term for this factor to be called lymphocyte-activating factor, or LAF. And I think I probably told you that when my lab used to say LAF was a joke, because it never really was able to get our T cells to grow, to proliferate. Anyway, they also then went on to say that the data that they published in this paper provides a very simple, reproducible, and sensitive assay. And that was true, and it was adopted by a whole cadre of scientists who became sort of a LAF clique of scientists, and they used this thymocyte proliferation assay as their baby. And they said that the LAF target cell is a purified T cell from either the thymus or from the periphery. And they further stressed that the LAF potentiated the responses to pure T cell mitogens, which really hadn't been established before. It had been established with antigens as well as for antibody production, but they hadn't shown it for pure T cell mitogens. And so that's one of the other things that they claim. they speculated as to, okay, so how does this do this? And they wondered whether it was a nutritional kind of thing, or maybe it was a hormonal kind of a thing, that the supernatant was just making the cells more sensitive to these factors that they needed, and if they were going to grow and proliferate and so forth and so on. But they didn't really have any other idea as to how that was. So that was the first paper. And then the second paper, they could have put it all in the same paper, but I guess maybe the journal made them break it up into two papers. If you're trying to build your CV when you're a young investigator, it's good because you get two papers instead of one. And so the second paper is called The Potentiation of T Lymphocyte Response to Mitogens number 2, the cellular source of the potentiating mediator. And this is by only Egal Gary and by Byron Waxman. For some reason, Gershon wasn't involved in those papers. And the data, and in the introduction, they basically state right up front, this paper shows data that LAF is produced by macrophages and by agents that stimulate these cells to make more of this LAF. And they used the same mice. They also did to look at the whole problem of whether or not it's a lymphocyte or a B cell or a T cell. They used exoradiation of mice, 850 rads, and then they would reconstitute the mice with either bone marrow or thymus or both to try to sort out the B cells and the T cells and so forth. And the other thing they did that you'll notice that it hasn't been used by the antibody people quite so much is that they used, instead of fetal calf serum, they used 10% pooled human serum. That's important because what we're showing in this whole thing is that, I'm getting ahead of myself. One of the things that they reference in this paper is the fact that LPS was shown by a paper that was in Press when they published their paper in 1972 by a group from the Basel Institute in Switzerland and also the Karolinska Institute in Sweden, that basically showed unequivocally that LPS stimulates the proliferation of mouse B cells. And then another paper that they reference, LPS does not stimulate the proliferation of human B cells. And I don't think we still understand that. I don't understand. Maybe somebody else does. If so, write me an email. So, in the results section of this paper, they made supernatants of human lymphocytes stimulated either with LPS or with PHA, and then they would test them on mouse thymocytes to potentiate the response to suboptimal doses of PHA, and that's their LAF assay. And they also tested mouse cells as well as rabbit spleen cells, mouse splenocytes and rabbit, and they both worked pretty much the same way, and that was sort of interesting. They also tested, they compared PHA and CON-A and LPS, and they said unequivocally LPS is the best stimulant for the production of this factor. When they took out, when they used purified adherent cells or got rid of the adherent cells, it was pretty clear that the major cell making this factor was the adherent cell, the macrophage. So, but then in the very first sentence of their discussion, they say that LAF is probably produced by macrophages. It seemed to me it was pretty much the data said it was, but they didn't want to, hang themselves if it wasn't. They said definitely and unequivocally the B cells were not a source, and they went on to say that LAF is probably playing a very essential role in all immunological responses where macrophages and T cells seem to cooperate. Then they enumerated all those different immunological phenomena that where they think LAF might be playing a role. One was in T cell proliferation to antigens and mitogens, which is what they showed in their papers. In number one, number two, in the ability of T cells to make cytokines. And one of the things that they referenced was the interferon response when PHA is used as a stimulant. But we also have blastogenic factor that was, published in 1965, 5 years prior to this, And they reference that, but they don't really talk about that. And they don't really talk about the fact that, so does one distinguish between the blastogenic factor and the lymphocyte-activating factor? And as time went on, we got all kinds of factors. We got T-cell activating factor, T-cell stimulating factor, and all kinds of thymocyte stimulating factor. And it wasn't necessarily clear whether everybody was talking about the same activity and same factor, or whether there were a whole bunch of different ones, essentially. Another one of the immunological phenomenon where they thought that LAF might be playing a role was in the mixed lymphocyte culture, which they felt was an in vitro surrogate for in vivo graft, allograft rejection. They also went on to say that this factor, this lymphocyte activating factor, or these macrophage activities may very well be one of the major mechanisms of actions of adjuvant. And I think that's very, very important. And at the time, nobody had a clue on how adjuvants were actually potentiating the whole immune response. So that's it. I think that we've covered most of the major points of this paper. And as time goes on, we'll see how this evolved into becoming interleukin-1, and then other factors became interleukin-2, and so forth and so on. So I'll stop there, and don't forget to like this video if you can, and click on the bell, and follow this journal club. We're getting all kinds of subscribers, by the way. So thanks. It's been fun.